We are in a climate emergency, and current industrial practices are increasingly unfit to deliver sustainable solutions. Ever-growing understanding of microbial systems have the potential to improve sustainability in various industrial processes. There is currently a disconnect between the science and the practical challenges of meeting the scale required to operate in these industries.
My group in the Plant Sciences Department at the University of Cambridge has established the Algal Innovation Centre, a facility which is designed to overcome the knowledge gaps and resolve the challenges associated with scaling of microbial production systems, particularly of microalgae. So to do this, we're currently working with a variety of industrial partners focused on activities ranging from food and feed to fuel and waste stream remediation.
We're helping our partners meet pressures of the modern marketplace through the development of sustainable processes that deliver functional products. Research from the Plant Metabolism Group, together with our academic and industry collaborators, aim to bridge the gap between theoretical potential of photosynthetic microbes and their actual performance at scale. Fundamental knowledge in cell and molecular biology, together with methods in analytical biochemistry, genome sequencing, and comparative genomics, have given our team a deep understanding of the microalgae that we work with.
We use our expertise to develop specific microalgal strains tailored to meet the bespoke requirements of a diverse industrial landscape. What do these products look like? We are helping pave the way for plant-based protein production, algal-derived pigments, vitamins, micronutrients, and omega-3 fatty acids, just to name a few examples. Harnessing our fundamental knowledge of the organism in question, we use a systematic approach at the lab scale to engineer strains with wide-ranging capabilities and properties.
Coupling a synthetic biology approach to streamline strain analysis enables us to identify factors preventing viable performance at industrial scale. For example, we have engineered algal species to express new pathways, developing strains that produce high-value medicinal compounds. Cycling these strains between the lab and the scale-up facility enables us to make the necessary changes to ensure high expression and overall productivity at scale.
Currently, there's no regulatory approval for engineered strains in the food and feed sector. However, we're engaged with regulatory bodies to help overcome this and pave the way for genetic engineering tools to be used to improve yields and sustainability in the food industry. In parallel, we're working with our partners to elevate the economic viability of scaled cultivation of GRAS status strains, that is, those that are generally regarded as safe, via conventional farming practices.
This primarily involves monitoring of macronutrient composition, for example, protein, lipid, or carbohydrates. These attributes are important for food and feed markets and can be incredibly variable depending on the cultivation techniques and conditions. There are numerous other performance indicators that we use to fine-tune cultivation procedures.
These indicators range from cell growth and viability, titer of production of compounds of interest like pigments, oils, and vitamins, to consistency and reliability of production of these compounds.
It has been the tradition in microbial cultivation to work with monocultures. This is because contamination has generally resulted in negative impacts on yields and performance. However, extensive work in our lab has given us a deep understanding of the many interactions between microbes, such as between algae and bacteria, and a recognition that favourable symbiotic interactions between these organisms are likely to realise the full potential of the production system.
In reproducibly tracking these performance indicators and paying close attention to dynamism that exists in nature, we help our partners evaluate their processes, strains, and products by way of life cycle assessment and techno-economic analysis.
Given our setup within the University of Cambridge, we are ideally placed to train the future workforce that is required to growth and development of the microbe-centric novel food, feed, and fuel sectors. So to this end, we run international training workshops, imparting our expertise in engineering, cultivation, and addressing challenges at all levels of the production pipeline.
Our future is being shaped by the global climate emergency. But here at the university's Algal Innovation Centre, we're fostering a network of academic and industrial collaboration. Together with our partners, we're developing sustainable solutions that will deliver real impact to the food, feed and fuel markets of tomorrow.